Once again, it is a great pleasure to be here to join you in this conference. And I thank the organizers for the kind invitation to join. So once again, I will be talking this afternoon about a particular organism. It's called Physarum, and a very interesting organism because it's multinucleated and it is a decentralized system. It does not have a brain, that is, but nonetheless, it exhibits directed migration. So as a physicist, what do we intend to learn from this? And also, I would like to show what are the problems that we are currently handling. I hope you can see these videos. These are two different experiments wherein you have also decentralized organisms reacts to stimuli. And usually in biology, they call this taxis, no? Taxis, like taxi, right? If it's due to chemicals, like the video in the left, this is called chemotaxis because there's a chemoattractant, okay? There's a chemoattractant, and so this dictee, they tend to go to the tip of the pipette. Years before, together with our group in Taiwan, we also do what they call thermotaxis, wherein we look at the migration of E. coli due to temperature gradient created by a laser, locally heating a mirror. Okay, so these are types of things that you see in biology, directed migration. But our interest this afternoon is about this organism called Physarum polycephalum. And as I mentioned, it's a very interesting organism. Back then, it has a big news because this Physarum turns out to be able to solve a lot of complicated problems. Like, for example, optimization of networks. And this is a good example, a video wherein the Physarum generates the subway system of Tokyo, okay? And in particular, in the experiments that we perform, we are looking at on the left side is the life cycle of Physarum. This one on the square, in the broken square, this is the part of a plasmodium wherein the Physarum is most active, like the one you see in the right side of the video. Okay. Okay. Now, why is it interesting? This Physarum, this plasmodium of Physarum, when it spreads out, they make this complicated network, okay, a topology composed of veins. And these veins, photoplasmic veins, they kind of intersect and which we call nodes, okay? On the right-hand side is actually a video we do in the lab, example of the things we do in the lab, wherein we look at the dynamics of these nodes, okay? As you can see, there is like pumps, okay? There are fluid moving from one side to the other. And we are interested. So these are in the kind of like the small scale. But one can look at this also in the bigger scale, okay? So there are two important questions that we look at here. The nodes that you see here, okay, what controls it, okay? So right now it's a big problem, open problem yet on this system. And then also to connect how the node dynamics leads to the organism-wide directed migration, which we have seen, for example, in the previous slide. Okay, so I would like to talk a bit of what we have done on those two questions, okay? So the first question is about direction, okay? What controls the node dynamics, basically? This is a part of a submitted work, okay? So our experiment follows the same format as before. The nice thing about organism-wide measurement is you don't have to do this in a microfluidic chamber with a very complicated setup, okay? So usually, you just have this petri dish and you put the favorite food of this Physarum, okay? And they tend to occupy this food source in time. Now, this is contrary for the case where you have no signal, okay? So the upper right video is for the case wherein you have no signal. As you can see, they just go randomly. But if you have a signal, they have this directed migration. So we would like to understand what causes this directed migration, okay? So the way to do it in our case is we would like to propose that we look at by the nodes, okay? As I mentioned, the nodes is something very interesting. And so we identify the nodes. So we take series of pictures of the video you see in the previous slide, and we look at where the nodes are, and by judging by the counts of how many nodes are there and how they are spatially distributed, we can extract some physics to it. Okay. This, for example, is the nodes, the spatial distribution of the nodes in these polar plots for different times. Okay, so this is like for 24 hours. After the left side, the left panel is after 24 hours, and the right panel is after 60 hours. So you see, the experiments last for about 3 days. Okay, in the video they seem to be fast, but actually it is we make it fast. Okay. So, but usually that video took us 60 hours to do, okay? So there are things that you do in biology to analyze this type of things. So in biology, they look at directional sensing, okay? They have a means to calculate that. They also talk about polarization, okay? And of course, this is kind of trivial. You can look at also node population dynamics. The fourth one is called motility, and motility has something to do with the force generation. You know, how was the traction generated? So, and unfortunately, well, I hope I still have time. That will be part two. I hope I can discuss that in detail later. Okay, so let's look at this. Well, one of the things you can see with this migrating physarum is the network structure, as I mentioned, right? And you can look at the connectivity, okay, which we call K. Okay, and as a matter of fact, you can look at the time evolution of K, and you look at the probability for those occurrence of K as a function of time. Definitely what you can see here is that the probability for three links is quite high. So a node connected by three protoplasmic tubes is higher than the rest. Although we also see something very interesting. Depending on whether the food source is near the physarum is near the food source, actually higher values of K, like K equal to 4 is expected. Okay. Now, like what can one can do is we can also do something like mean field approximation of the nodes, right? One can do weighted average of the node's position, like a centroid, okay? And one can calculate the centroid, and then one can also plot it, right? So we did that here in this plot in the middle, and this is the centroid of the nodes, right? With respect to the food source. So Fs is like the food source. So food source 1, 2, and 3. As you can see, the physarum actually moves in a very interesting, it's really directed migration. Okay, so it goes to food source 1, and later on it chooses food source 2 and 3. By the way, you might be wondering whether this is only true for one realization. As a matter of fact, we did this to many realizations. Okay, yeah, but not all the time they get this counterclockwise direction, right? Sometimes they do the opposite, but you always get kind of the same behavior wherein they have directed migration. They go from one food source to the next. And for this particular case, because it's counterclockwise, we kind of choose a parameter, which we call phi, to look at how it changes in its in polar position as a function of time, and more like a directional cosine. And one can do is that if it's one, then basically this two, it's actually going to the counterclockwise direction. Okay, so as you can see, we find that about 70% of the time it actually go in a preferred direction, right? In this counterclockwise direction. But there are some times that it stop, okay? And this occurs mostly when it's in the food source. So if you consider that as a means of correct sensing, then for this system, it about 90%, okay? So 90% accuracy in the track. So this physarum is very good, okay, in finding the food, okay, and this actually correlates with previous experiments, especially with the links, okay. Now the another thing that we are interested, which biologists usually do, is what they call polarization, and polarization has something to do with the morphology, the body shape of the organism, okay. For example, for dicty, it will be like how the dicty is either stretched or a bit circular, okay? So we did that also here, with this time using the nodes, but this time we calculate, like embed it like in an area. And I think for the statistical mechanics people, one can look at this as the Lewis statistic area, okay? So which you bound by the outermost nodes. These are some the pictures in the middle of the LSA, the Lebesgue spastic area, as a function of time, wherein the plus, on the other hand, is the food source. Okay, so we are interested about the shape, the morphology of the physarum as a function of its distance with the food source. Okay, so we did a simple analysis of this LSA by fitting it with an ellipse. Okay, so you can define an W and L, and you can take the measure of this W and L by taking the ratio. Now, obviously, if it's circular, then W over L will be 1, right? But if it's not, then it will be higher values. And so we did that over here, the plot in the bottom, wherein we look at the distance between the nodes centroid with respect to the, the W over L with respect to the node distance to the food source. And we find something very interesting. As long as you are quite near to the food source, the shape of the nodes is actually more or less circular or close to 1. Okay, although there are, of course, there are some data points still above that by about 20%. On the other hand, when you are farther from the food source, most likely you have higher values of this ratio of W over L, which we call a polarized state. And it happens is that this polarized and depolarized states actually changes as the physarum nodes goes near or out of the food source. Okay, so this is an interesting finding, and as a matter of fact, we find something very interesting is because if you look at this transition of the food source, of the polarization and the polarization of the physarum morphology, it actually corresponds to this excitable nature of the number of node counts. Okay, so the circle is the experimental data. Okay, and the red one is just a very simple, the typical Fixed-Nagumo model, and which you can fit. Okay, just to train the eye. And our interest here is precisely on the experimental data. And as you can see, this number of nodes, the one with the symbol, takes some time, and then it goes up, and then it rapidly goes down. Okay, and then it goes up again, rapidly goes down, as it moves from one food source to the other. Okay, so these are interesting because I think this signifies a clear manifestation of an excitable dynamics. But unlike that in neurons, here the threshold is not necessarily 0. And the main reason for that is because the physarum needs also to feel the environment, especially the chemoattractant field. And and again, this correlates with this polarization, depolarization transition. Okay, so at maximum number of node counts, the physarum is polarized. And after that, it becomes depolarized. And again, it goes polarized and then depolarized. Exactly with the same dynamics with the node counts. Okay, so, before I go to the simulation, what does it tells us? Well, it tells us a bit about the property of the node. We think that this type of dynamics tells us that there must be at least 2 thresholds, okay? One that controls the creation of the nodes, and one that leads to the kind of annihilation in the physics term of the nodes, depending on the intensity of the chemical signal that a node is getting, right? So in that sense, you can picture out the nodes, as a matter of fact, like sensory components of an oscillator device. And we did want to check that, and in the minimum, can you use 2 thresholds? Okay, can we do 2 thresholds for a simple node to generate the spike dynamics that we see in experiments? So we did that. We did Brownian lattice simulations. First, we kind of generate the this number 1, the signal intensity field. One can solve the diffusion equation with decay rate, and one can do that easily. And then you can generate into a lattice. Next is you kind of put like, how do you call it, an inoculate, okay? So you start with a seed, and then you allow the nodes to propagate, okay? in time by some very simple rules. Okay, the first one is that the next lattice to be changed in its node composition, the distance from the previous node is not necessarily uniform. Okay, it's given by this R, where R is a function of the chemical intensity of the seed node. Okay, so here, as you can see, this is a Hill function of order 2. And we also put stochasticity here by putting this noise. But this noise, eta, is white noise. Okay. Well, in this case, in the figure, what you can see is that you can see this jumping. Okay, assuming it's 1, you can see that it can jump to the next lattice site, and there are 9. How many here? 8 possibilities. Okay, I'm sorry, 9 possibilities, including its own. And then what you do, these are now the sites that can be updated, and then we ask whether the computer will allow the next to the right of this node, red, to be updated. Okay, well, it can only be done if the intensity of that node is bigger than some threshold delta. Okay, so this is threshold number 1. So, and this is about creation. So a new node is created as long as the intensity of that new lattice site is greater than the previous lattice site by a parameter delta. Okay. On the other hand, the previous node can also be annihilated. Okay, it can be annihilated if its previous intense chemical signal intensity is now given by this parameter epsilon. Okay, so these are the 2 thresholds. Again, you have delta and epsilon. So and then you do this iteration for until the food source is totally visited by the physarum. I think this is okay, right? Good. So this one in your left is the video I showed you earlier, data, and the one on the right side is the simulated data we did with this Brownian lattice simulations, and yeah, with these parameters over here. And beautifully, I think it captures the dynamics that you see on the left side. Okay, and as a matter of fact, the same types of observations can be observed, even especially in the spike. Okay, the spike dynamics in the node counts and its relation to the polarization depolarization dynamics. Okay, they are also coupled. Okay, so we see this excellent agreement. So we are happy. So I think this is a very, it's a simple experiment that gives us a lot of understanding about the importance of nodes as far as this organism is concerned. Okay, and that the next step would be to go deeper on our understanding of this and to look at the chemical pathways, specific chemical pathways that that yields one that we have observed. Okay, okay. So that's part 1. So I don't have a lot of time. So I go to part 2? This is also very interesting because here we are interested about the nodes themselves, the dynamics of the nodes, and how it leads to directed migration. Okay, so let me clarify things first. Okay, so there are different time scales and length scales in this system. Okay, so the one in the left side is the node itself. Okay, so we're also studying that the scale is about 200 microns or smaller. Okay, you can go up to 1 micron or even smaller than that, about 10 microns over here. Okay, and the next time length scale is the network itself. Okay, the protoplasmic network, and this is about a millimeter. And then later on, you have the whole organism moving to this. It's about 1 centimeter. Now, there are different forces yielding from different time scales on this one. Okay, you have protoplasmic streaming force, and you also have destruction force generation. Okay, so we are interested with also measuring these forces. So in our lab, we measured these different scales by different methods. Okay, especially for the small one. Jay Lord, a PhD student of mine, is using optical tweezers we set up in our lab to measure these things. Okay, so you use light to capture a bid or an organelle, and then from the fluctuations you can measure the force. Another thing that we do in the lab is we also do destruction microscopy, wherein you have these micropillars, and then by looking at how these micropillars behave as with this physarum, you can estimate the force. But what I will be showing you later is what they call passive traction microscopy. wherein we just use these beads embedded in a gel where the physarum is in it. And then by looking at the properties of the fluctuations of the beads, we can elucidate the force. Okay, so do I still have time?
Could you please summarize the remaining part of your presentation?
Okay. Yeah, okay. So too bad, but definitely one of these days we can discuss if you are interested. Anyway, I think the most the most important part is that for this study, we were able to capture the 3D migration for the first time of physarum, and we were able to map it. This is a good example of experiment where we're able to look at how these oscillations create this 3D dynamics. And we now have a new proposed model for this system. And not only that, these fluctuations of these beads, because of this physarum, can actually be reproduced also by a modified active trap model. It is basically this model. You have inertia term, you have this dissipation, and you have this active force, which is just given by this term over here. Okay. Anyway, I summarize that. By saying that we have performed experiments and we have inferred from experiments and also from simulations the 3D migration of the physarum, and we validate some of the earlier models of this and even extend it now to a 3D. And these non-equilibrium fluctuations are very rich. I think these are one of the first systems that has shown oscillatory components in the fluctuation. So, and the trap model, we were able to modify it, and it yields also good results. Okay, so thank you so much for listening. Yeah, thank you.
Good afternoon. My name is Yoshitada Morikawa, and today I will introduce our theoretical work on carbon dioxide hydrogenation on kappa surfaces. In our group, we investigate materials which are important in economy, energy, and environmental problems. And we would like to solve the trilemma by using quantum simulations. And especially, our group is focusing on interface problems, because interfaces play very important roles in wide variety of applications, such as organic devices, or let me see, organic devices. So in organic devices, the electron and hole injection barriers at metal electrode and organic semiconductor interface plays a very important role. And quite often, it is difficult to clarify the electronic structure and atomic geometries at the interface, because this interface is embedded, buried in the under the material. So first principles simulations play very important roles. And also in fuel cells, the efficiency of fuel cells depends on the chemical reactions at the electrode-electrolyte interfaces. And in these fields, first principles theoretical simulations play very important roles. And our group, has been focusing on interface problems, especially organic metal interfaces. We investigated thiolate metal interface. This is famous as self-assembled monolayers, and we investigated atomic geometries and the bonding nature and also dynamics of thiolate on metal surfaces. And also we investigated the interface dipole formation at the organic metal interface. This is a typical physisorption system, normal alkane metal interface. And this is a pentasene metal interface, and this is ALQ3 metal interface. These are famous for organic devices. And also we investigated magnetic molecule metal interface. This is important for spintronics devices. And also we have been intensively working on heterogeneous catalyst. This is NO dissociation, the NO dissociation on metals. This is important in three-way catalyst. But in conventional three-way catalyst, platinum, rhodium, and palladium precious metals are used. They are quite expensive and expensive metals. Therefore, it is desirable to reduce the usage of those precious metals without reducing the reactivity of catalyst. And copper is one of the promising candidates to replace precious metals in three-way catalyst. And we investigated that NO dissociation, usually it is possible on rhodium surfaces, but the copper, usually barrier is high, but we clarified that the water hydrogen bond from water can enhance the dissociation of NO dissociation on copper surface. And also we investigated the suppression of sintering by perovskite substrate. And also we investigate hydrogenation of carbon dioxide to methanol. I will explain in more detail of this mechanism. And also we investigated the reaction process of etching of silicon carbide using platinum catalyst. So silicon carbide is a promising, important material for power devices. But silicon carbide is mechanically very hard and chemically rather inert. Therefore, it is difficult to etch the surface. But the experimental group in our department succeeded in fabricating atomically flat silicon carbide surface by using platinum catalyst. So we investigated the etching process at platinum, water, silicon carbide, three-phase interfaces. And also we investigated the enhancement of water dissociation on thin film oxide. And also we have been intensively working on electrochemical processes. This is a hydrogen evolution reaction at the water metal interface. This is a typical, most typical electrochemical reaction. And in our simulation, we included solvent effect and also electrode potential explicitly into our computational model. And also, this is not a chemical, electrochemical process, but a homogeneous catalyst, the ligand-free Suzuki cross-coupling. We clarified the chemical process, chemical reaction process. And this is methanol oxidation at the water metal interface. This is important for direct methanol fuel cells. So, and today I will introduce our work on hydrogenation of carbon dioxide, especially vibrationally enhanced hydrogenation of carbon dioxide for methanol synthesis. And this is done by our former PhD student, Paji Mutaki, when he was in our group. And the methanol is synthesized from a gas mixture of carbon monoxide, carbon dioxide, and hydrogen using copper zinc oxide catalyst under 250 Celsius degree and 5 megapascal. And from isotope experiment, it was concluded that the carbon source of methanol mainly comes from carbon dioxide. Therefore, carbon dioxide is directly hydrogenated to methanol. But this process is rather slow. The first hydrogenation of carbon dioxide to formate is rather slow process. And from experimental and theoretical work, previous experimental and theoretical work, we concluded that this process should be irreducible mechanism. In surface chemistry, there are two mechanisms. One is Langmuir-Hinshelwood mechanism. In Langmuir-Hinshelwood mechanism, adsorbate and adsorbate react. But in irreducible mechanism, the gas phase molecule directly attacks the adsorbed species, and the chemical reaction takes place. So we investigated the energetics of this process using density functional theoretical calculations. Here, energy profile is shown. And we used several energy functionals, the ordinary conventional GGA, PBE, or dispersion corrected PBD2, and recently proposed van der Waals density functionals. The conventional GGA cannot describe the physisorption state of carbon dioxide. But recently proposed van der Waals density functionals can reproduce both physisorption and chemisorption processes rather well. And therefore, our calculation is consistent with the experimentally observed activation barriers for synthesis process and also for decomposition process. So for synthesis process, the energy barrier is experimentally measured to be about 0.6 eV, and our calculation using recently proposed van der Waals density functional agree quite well. And also decomposition process, the barrier can be comparable with the experiment. So our calculation reproduce this energy profile rather well. Then, as I mentioned, the first hydrogenation process of CO2 to formate is an irreducible mechanism. And this suggests that there is a possibility to enhance the hydrogenation of carbon dioxide by controlling the energy state of impinging carbon dioxide. And this must be related to the reverse reaction, namely decomposition of formate to carbon dioxide. So the elucidation of the dynamics of formate decomposition is important for improving catalytic methanol synthesis. And therefore, this experiment was performed by a group of Tsukuba University, and this is the result. So this figure shows the energy and orientation of dissolving carbon dioxide from decomposition of formate on copper surface. And it turns out that carbon dioxide desorption mainly takes place towards the normal surface normal direction. And also the translational kinetic energy of dissolving carbon dioxide is about 0.1 electron volt. And this is independent of the surface temperature. So this means that this process is not in thermal equilibrium process. And also the translation energy of dissolving carbon dioxide is only 0.1 eV. This is much smaller than the activation energy of formate synthesis. So the question is, how about the rest of the energy? So we have performed first principles molecular dynamic simulations of carbon formate decomposition, and we investigated the energy profile of dissolving carbon dioxide. So this is the energy profile as I shown, and So starting from this transition state, we investigated the dynamics of the decomposition process. So from transition state to initial state is a gas phase carbon dioxide. So carbon dioxide dissolved from the surface. So starting from this transition state, we investigated the kinetic energy profile.
Please wrap up your presentation. Yeah, 1
yes.
minute. Thank you.
Yes. And we analyzed the energy profile. And it turns out that the dissolving carbon dioxide, the OCO bending mode is significantly enhanced. And so about half of the activation energy is transferred to OCO bending mode. And the origin of the OCO, the excitation of OCO bending mode can be understood by looking at the transition state. At the transition state, the OCO angle is changed, and then CH bond is broken. So upon decomposition formate, the OCO bending mode is strongly enhanced. And this suggests that by exciting OCO bending mode of gas phase carbon dioxide, the formate synthesis should be enhanced. So this is our theoretical simulation. So gas phase carbon dioxide, the OCO bending mode is excited, then hydrogenation process is must be enhanced. And recently, this enhancement of formate synthesis by excitation of CO2 vibration has been observed experimentally, and we have published joint theoretical experimental paper. And this is the summary of our presentation. Thank you for your kind attention.
So I'm here to present our recent results entitled Tip-Enhanced Resonance Raman Spectroscopy and Imaging of a Single Molecule. So more details of our work can be found in our recently published work. So Raman spectroscopy is a powerful technique for studying different kinds of materials. However, it has some disadvantages. For one, the Raman cross-section is very weak. It's about 10 to the 14 times smaller than fluorescence and about 10 to the 10 times weaker than absorption spectroscopy, making detection of Raman signals very difficult, especially in the presence of fluorescent signals, as seen in this figure. Moreover, the applications of Raman spectroscopy to nanoscale materials is quite challenging owing to the diffraction limit, which limits the spatial resolution of Raman microscopy to about half the wavelength of the incident light. So in order to solve these two problems, one technique that can be applied is so-called tip-enhanced Raman spectroscopy, wherein we use the tip of a scanning probe microscope such as STM or an AFM in order to enhance the Raman signals and therefore improve the spatial resolutions. And in fact, tip-enhanced Raman spectroscopy, specifically STM-based TERS, has been applied successfully to different kinds of materials, such as carbon nanotubes, wherein in ambient conditions, spatial resolutions as high as 1 nanometer was obtained. And also, it was successfully applied on single molecules in ultra-high vacuum and low temperature conditions, first reported in 2013. And more recently, it was shown that STM-based tip-enhanced Raman spectroscopy can be used to identify individual vibrational modes. For conventional STM-based TERS measurements, the molecule is placed directly on metal surface. However, the interaction between the molecule and the metal surface complicates the analysis of such a system. So in order to avoid this problem, we show in our work, we utilize a sodium chloride surface which acts as a decoupling layer in order to separate physically the target molecule and the metal surface, and then we can perform STM TERS. Moreover, aside from STM TERS, a variety of measurements can also be applied on the same sample, apart from just Raman spectroscopy. So for our work, we study a single copper naphthalocyanine molecule on sodium chloride supported by silver 111 surface. So what we do is we deposit our copper naphthalocyanine molecule on both metal surface and sodium chloride surface on silver 111. And then when we approach the tip on the STM tip on the molecule, we found that the Raman signals appear only when the molecule, when the tip is near the molecule, and when the tip is placed far away from the molecule but still near the sodium chloride, all Raman signals are gone. This suggests that we have successfully shown that the Raman signals are greatly enhanced. More interestingly, actually, is that when we place the tip near the molecule on the silver surface, no Raman signals were observed, wherein using the same tip under the identical conditions, TERS signals are observable on the molecule on sodium chloride. In order to understand this, we studied the electronic structure of the molecule by using scanning tunneling spectroscopy. So we performed STS on the molecule on sodium chloride and on the molecule on silver 111. We observed clear, sharp peaks, both on the positive and negative side of the bias voltage for the molecule on sodium chloride, wherein very broad peaks were observed for the molecule on silver 111. Moreover, when we adjust the STM scanning conditions, we have found obvious change in the STM topography of the molecule in the in-gap state and on the LUMO of the molecule. And in fact, by comparing with DFT calculations, we know that when we scan the molecule using STM at this bias voltage, we can observe different HOMO and LUMO topographies, and these topographies nicely match to the DFT calculated molecular orbitals. These results clearly shows that the molecule is electronically decoupled from the metal surface, unlike when the molecule is on the silver 111. Then the implication of this for the Raman spectroscopy, and specifically for tip-enhanced Raman spectroscopy, can be understood when we look at the enhancement mechanisms in both SIRS and TERS in our case. So for tip-enhanced Raman spectroscopy, the Raman signal is enhanced by two or three kinds of enhancement. First is the electromagnetic enhancement, wherein both the incident light and the outgoing Raman light is enhanced by the presence of the metal tip. However, this enhancement cannot explain the absence of Raman signal for the molecule on silver, because in this experiment we use the same tip. On the other hand, another source of enhancement is so-called charge transfer or chemical enhancement, wherein the excitation happens from the Fermi level of the metal surface and the molecule. But in our case, according to STS measurements, the molecule is decoupled. So because the STS measurements don't show any hybridization, therefore we can say that the chemical enhancement is suppressed in our experiments. So the other possible source of enhancement is the enhancement by the excitation laser under resonance conditions. In this case, we believe that the molecule is undergoing a direct molecular resonance, that is, the direct transition from the HOMO to the LUMO is obtained by adjusting the energy of the laser. And we were able to do this by utilizing a tunable laser. Now, in order to do this properly, we have to identify the absorption of the molecule. So where is the absorption, molecule's absorption? So fortunately, in the single molecule case, our group has developed a technique in order to that can measure the single molecule absorption reported in this paper in 2017, wherein we use the localized plasmon generated at the tip metal junction and use it as a kind of light source as it is used in the conventional absorption spectroscopy. And then this localized plasmon light, the light produced by this localized plasmon is absorbed by the molecule. And then our member was able to show that the transitions, the sharp transitions are corresponding to the absorption of the molecule. Then we applied this technique to our system. And then first, when we look at the molecule on silver, we have found that under the wavelength range used in our experiments, no absorption is observed. This suggests that resonance enhancement is not possible for molecule on the silver 111 surface. Meanwhile, for the molecule on the sodium chloride, we have observed a clear dip which corresponds to the absorption and the transition between the homolume of the molecule. And when we compare to room temperature absorbance measurements measured using UV visible measurements, the absorption peaks are quite matched. So now that we know the absorption of the molecule, we tried to vary the excitation wavelength of our laser in order to observe the resonance Raman effect. So what we did is we changed the wavelength of the excitation laser under in this wavelength range, and then we observed the individual Raman signals. And then when we plot the vibrational peaks, we can observe such a plot. Here we can see that the absorption measurement, the absorption results nicely matches the Raman signals of selected peaks. And in fact, all the peaks follow the same trend. This is a clear evidence of direct resonance Raman effect at the single molecule level, which has not yet been shown previously. Under these resonance conditions, we also tried to perform Raman mapping, wherein we measured the Raman signals of our molecule by placing the tip on around the molecule. So when we perform the Raman mappings and then we look at certain peaks, for example, this peak, we observe such a Raman map. So the Raman signals are intense at this circular region. On the other hand, for example, in this peak, we found that the Raman signal are only strong at these 4 positions. Lastly, for this peak, we found that the Raman signals are only strong at these 4 positions. And when we check all the Raman maps with good signal-to-noise ratios, we were able to find that there are only 3 patterns of the thirst maps: circular, cross, and plus. In order to understand what is happening here, we looked at the Raman maps individually. First of all, for this kind of cross map, when we compared with DFT calculations, we found that these maps, these modes, for example, the 1389 mode, has a symmetry of B1G. That is, the vibrations are symmetric along this symmetric axis, along this axis. So the vibrations are symmetric along this axis and this axis. And so when we considered the position of the tip under the thirst measurement, so for example, when we place the tip here, the vibrations are in phase to each other. The vibrations around the tip is in phase. Meanwhile, when we place the tip here, the vibrations are out of phase. So considering this, we can clearly see that Raman signals will be enhanced at the positions of the symmetric axis. So in the case of B1G mode, it will be around this cross. On the other hand, for this mode, we found by when we compared with DFT calculation that this is the B2G mode. This kind of mode has a symmetric axis along this axis, and considering that we put the tip around these positions here, at these positions, the vibrations are in phase, while here the vibrations are out of phase. And then we can observe such a vibration wherein strong Raman signals are observed at the plus position. On the other hand, the 680 mode is a A1G mode. It's basically symmetric at all directions and in all of those directions. So it's basically enhanced everywhere. So when we check all our Raman maps, we can classify them according to the symmetry. So A1G is the circular, and then B1G is the cross, and then B2G is the plus. So our observations suggest that the vibrational symmetry can be revealed by STM Thurs mapping under direct molecular resonance conditions. So in summary, we have observed resonance Raman spectroscopy at the single molecule scale, and then we were able to spectrally and spatially we have a spectrally and spatially resolved Raman maps of a single molecule, which revealed that the vibrational symmetry governs the patterns of the Raman maps. So acknowledgements, our group. I would like to acknowledge our co-authors, Professor Kim, Dr. Imada, Dr. Hayazawa, Dr. Yang, and Dr. Kasama. And then the theoretical support is provided by Dr. Miwa from Northwestern University and Mr. Takenaka and Professor Iwasa and Taketsugu from Hokkaido University. So I'd like to acknowledge our funding from RIKEN's Program for Junior Scientists and from Kakenki. Thank you.
Hello, I am Dr. C.D. Mata, and today I will be presenting our paper entitled Fourier Transform Infrared Spectroscopy of Nd:YAG thin films by femtosecond pulse laser deposition with nitrogen and oxygen as background gas. First, we will define thin films. Thin films are a layer of materials deposited on a substrate. This material thickness ranges from a fraction of nanometer to several micrometers. Thin films have a wide range of applications, and some of these applications may be encountered in everyday life, such as thin film as optical coating, which uses the anti-reflective coating that reduces reflection of optical surfaces such as photographic lenses. Another application is thin film in a semiconductor. These tiny devices made from the interconnection of different thin film layers. Another application is in solar cells. Thin films play a role in the coating of the solar collector, which enhances the photothermal conversion efficiency. Now we move on to the process of producing thin films, which is the pulse laser deposition. This is a physical vapor deposition technique that uses high-powered pulse laser beam to ablate the target. PLD is a common way to produce thin films due to its confidence of stoichiometrically transferring the target to the substrate. This is a vacuum chamber with the target attached on the target holder and the substrate on the other end. When a laser emits optical pulses, energy is transferred to the particles on the surface of the target that results to the ejection of highly energetic particles, which creates the plasma plume. These ejected particles travel its way from the target to the substrate and condenses on top of the substrate. In this experiment, we use a femtosecond laser which emits optical pulses in the domain of femtosecond. This produces highly energetic particles. These highly energetic species ablated from the target may cause damage to the surface by sputtering and may cause defect formation in the deposited film. The introduction of background gas eliminates this phenomenon by the addition of collisional processes. The kinetic energy of the ejected particles is reduced upon interaction with the background gas, and this allows the growth of particles with low energy on the substrate. Several parameters affect the properties of deposited materials, which include the type of background gas used as well as its pressure. The growth behavior of deposited materials in many physical vapor deposition systems can be determined using the free energy per unit volume of the vapor, or delta G sub V, given by the equation, where K is the Boltzmann constant, T is the absolute temperature, omega is the atomic volume of the film atoms, P is the pressure of the incoming ablated atoms, and P sub V is the vapor equilibrium pressure. To relate delta G to the background gas pressure, we set P is equal to epsilon P naught, where P naught is the inherent pressure with the absence of any background gas, and epsilon is a positive valued constant that relates the background gas pressure P to P naught. Combining this equation, we will get a semi-quantitative behavior about the dependence of delta G sub V to the varying background gas pressure as seen in the equation. For constant substrate temperature experiment, the change in the free energy per unit volume decreases as the coefficient of epsilon increases. Since delta G dictates the growth mechanism of the film, it can predict the resulting properties of the film. An increase in the delta G results to an increase in the total free energy barrier, and this promotes monolayer growth of the film, while a decrease in delta G results to a decrease in the total free energy barrier that leads to either or both nucleation and island growth. In this paper, we analyzed the effect of nitrogen and oxygen as background gas for deposition of NdL thin films on glass substrate using femtosecond pulse laser deposition with the use of FTIR spectroscopy. This is the FTIR analysis of the deposited films under the influence of oxygen and nitrogen gases. For the 50 milliliter O2, the black line on the graph shows a 24% transmission on the peak around 980 and 79 transmittance for 1,400 peak. These peaks were also present in the film under 50 milliliter nitrogen. The peaks under the influence of nitrogen exhibited higher transmittance with 31% and 85%. Additional peaks were also identified on 2,200 and 2,560 with 96% and 97% transmission respectively. Nitrogen as background gas promotes preferential growth with continuous and smoother morphology. For deposited films under oxygen as background gas, lower substrate coverage can be attained. A possible reason for this is that rougher film with higher porosity was also produced due to the interaction of ablated species and oxygen. Since oxygen is larger than nitrogen, the reduction of kinetic energy through collision was much more effective for oxygen. And this much reduced kinetic energy resulted in sputtering instead of deposition, hence low coverage under oxygen gas. The pressure of nitrogen was also increased to 80 millitorr, and the FTIR analysis for this is shown in blue line. These peaks were similar to those of the films deposited under 50 milliliter nitrogen. The 980 and 1,400 peaks shows a 28% and 84% transmittance, and for the other two peaks, the transmittance was 92% and 94%. All of the peaks for 80 milliliter manifested a lower transmission as compared to the films under 50 milliliter. Further increasing the pressure of the background gas increases the coefficient as seen in the equation. Increasing the pressure will lead to an increase in the epsilon, and this decreases the delta G sub V, which promotes coalescence, which can be seen as the lower transmittance of the film. So in summary, 3 films of NdAg were deposited using femtosecond pulse laser deposition under the influence of varying pressures of nitrogen and oxygen. The deposited films under oxygen exhibited lower transmittance as compared to the film with nitrogen background gas of the same pressure. Film under nitrogen yielded an overall higher transmittance. On the other hand, increased surface roughness produced by the interaction of ablated materials and oxygen causing sputtering can explain the lower transmittance of the optical energy on films deposited with oxygen background. And lastly, increasing the pressure of nitrogen background gas, which can be explained by a possible increase in surface roughness of thin films, lower the transmittance. We would like to acknowledge the Natural Science Research Institute for this project, and all research work was conducted at the Photonics Research Laboratory at the National Institute of Physics, UP Diliman. Thank you.
Good evening. I will be presenting our work entitled X-ray diffraction analysis of strontium-doped lanthanum cobaltite powder prepared from an aqueous precursor solution. To start, lanthanum cobaltite is an insulator which is rhombohedral perovskite in nature. But when strontium-2 plus is substituted into the lanthanum site of this perovskite, the lanthanum cobalt oxide insulator becomes a ferromagnet. This ferromagnetism observed in this material can be explained by the oxidation of the cobalt array, which changes lanthanum cobalt oxide into an electrically conducting LSCO. It can be said that the strontium-2 plus substitution yields a remarkable improvement in the properties of the lanthanum cobalt oxide. However, it is difficult to form high-purity LSCO powder using conventional methods. The most common method of producing LSCO powder is the conventional solid-state reaction method. In the solid-state reaction method, constituents metal oxides and carbonates are mixed and are annealed to very high temperatures for several hours. In addition, the problem with the solid-state reaction method is often the formation of significant amount of impurity phases. These impurity phases are possibly a result of inhomogeneous mixing of the precursor powders due to large particle sizes. The particle size from a mechanically milled precursor powder can only go down to around 0.5 to 1 microns in diameter. One can further decrease the particle size of the precursor powder by using a planetary ball mill. In this way, milling can only produce particle sizes that are about 100 to 500 nanometers in diameter depending on the milling parameters. In this study, a simple route is investigated for the preparation of LSCO powder using an aqueous precursor solution. In this method, we eliminate the mechanical milling step that is done in the conventional solid-state reaction method. This method uses metal acetate and a complexing agent mixed in an acidic solution in order to eliminate the need to perform mechanical mixing. Metal acetates, when mixed in an acidic solutions, are converted into metal ions. Metal ions are then complex by an organic ligand which prevents precipitation of the precursor solution and allows homogeneous mixing of the precursor solution. For the experimental details, lanthanum acetate hydrate, strontium acetate, and cobalt acetate tetrahydrate were dissolved in 50% acetic acid and DI water solution. To form the complex, EDTA was first dissolved in an ammonia solution, then mixed into the metal acetate solution. Two sets of samples were prepared, one with pH of 5 and one with pH of 7. After mixing, the precursor solution was dried at 150 degrees Celsius for about 4 hours until dry, pyrolyzed at 350 degrees Celsius for 20 minutes, then finally heat treated at 700 to 850 degrees Celsius for 20 minutes. All our samples are characterized by powder X-ray diffraction. This shows the XRD pattern for our samples prepared at pH equals 5 and is heat treated at 700, 750, 800, and 850 degrees Celsius. XRD shows that most peaks belong to the LSCO phase, with minor impurity peaks that are around 2 theta equals 30 to 40 degrees. The suspected impurity phases were lanthanum oxide, strontium carbonate, and cobalt oxide, which are usually observed in LSCO prepared using the conventional solid-state reaction method. Rietveld refinement method was used to calculate for the lattice parameter and the volume fraction of the LSCO powder prepared from pH equals 5 solution. Calculated lattice parameters show that there is no significant change in the unit cell size for all heat treatment temperatures used in this study. The calculated volume fraction shows that most of the samples prepared are LSCO dominant phase. Moreover, there's a slight increase in the volume fraction of the LSCO as we increase the annealing temperature, while a slight decrease in the strontium carbonate phase. It can be said that the increase in heating temperature possibly decomposes the strontium carbonate phase, then reacts with the other metal oxides to form LSCO. Another set of samples was also prepared to determine if we can reduce the impurity phases that are present in the previous samples by increasing the pH neutral. This time, the pH of the precursor solution was adjusted to 7.0. The XRD data shows most peaks also belong to the LSCO phase with the impurity peaks around 30 to 40 degrees. Similar suspected phases were also considered during the Rietveld refinement method. Lattice parameters and volume fraction were also calculated from the powder XRD data. It can be seen that there is no significant change in the unit cell size for all annealing temperatures. The calculated volume fraction also shows that most samples prepared were LSCO phase dominant. Now, we compare the LSCO yield and the amount of impurity phases obtained from the two sets of samples. We see that there's a slightly more LSCO yield in the set of samples prepared at a more acidic pH. This is also consistent with the decrease of the strontium carbonate phase. However, due to large uncertainty of the volume fraction of LSCO, we see no significant effect of the pH the resulting LSCO phase yield. This is also true with the effect of heat treatment temperature. As a summary, we present a simple route to prepare strontium-doped lanthanum cobaltites using an aqueous precursor solution. Our result shows that all the powder samples contain about 85% LSCO with a small amount of impurity phases. Impurity phases that are found were lanthanum oxide, strontium carbonate, and cobalt oxide. Annealing has no significant effect with the resulting LSCO phase yield. Further optimization is necessary to produce high purity, which is about greater than 95% LSCO powder. Thank you very much for your attention.